Welcome to Bio 205 Microbiology with Pima Community College. In this video, we're going to show you how to collect bacteria from natural sources, especially fomites, and we're also going to collect bacteria from the air. So before we get started, the materials you will need are your lit and ready Bunsen burner. We're going to use a glass tube of sterile water, our test tube rack. We're also going to use a sterile cotton swab. We have 2 Petri plates, and our fomite today again is going to be the knob on our soap. So what you first want to do is label your Petri plates as follows. On one plate, we are going to collect bacteria from the air. So remember, we're going to write around the edge of the Petri plate. I'm going to write air sample, today's date, my initials. The type of media we're working with in this lab is called nutrient agar, so I'm going to initial this NA for short. We're actually going to incubate these since these are coming from our environment at 25 degrees Celsius, as close to room temp as we can achieve. For our second Petri plate, we're going to indicate that this is coming from the fomite, and specifically, since we don't know what we're going to collect, we're just going to call it soap knob. Again, repeat your initials, the date, type of media, and your incubation temperature. So for your air plate, this one's super simple. What you guys would normally be doing in class here would be opening the Petri plate. We're going to move it farther away from the Bunsen burner. We're going to remove the cap, and we're going to see what contamination this Petri plate will collect from the air. And we're going to let this sit anywhere from 5 minutes up to 30, depending on how long you'd like to let it be exposed. So for our second Petri plate, our fomite, what we're first going to do, using our aseptic technique, remove our sterile cotton swab. We're going to pick up our glass test tube of sterile water here. Remove the cap with my little finger from my dominant hand. Remember to pass it through the flame. And we're just going to dip the cotton swab to moisten it. This is going to help us pick up more bacteria from the surface. Replace the cap and place down. Now from your chosen fomite, we're going to take the cotton swab and we're going to rub over the surface, going in a circular-like motion. We're going to go to our Petri plate, pick it up, and we're going to perform what's called a lawn inoculation. For this lawn inoculation, we're going to rub the surface of the swab, rolling in a roll-like fashion, down the center, and then I'm going to repeat, going in the opposite direction. I can rotate the Petri plate with my hand, checking to make sure that I'm covering the entire surface. You can continue to rotate your Petri plate and rub your swab all over. Once the entire surface of the Petri plate has been inoculated, replace the cap. Your paper here is just going to go in the regular trash, and your swab is going to be disposed in typically biohazard. This has only been touching surfaces in the room, but that's typically where we're going to place these as biohazard. So incubate your Petri plates and come back and check to see what grows. To give you an example, this would be representative of a fomite in the classroom. We're going to examine photos of this in lab later, virtually, and you guys will get to explore what all of this growth is and how to identify different bacteria growing from your fomite.